Hey team, Dr. Jack Gordy, and in this video I'm going to be covering how old are your tissues. Is your skin older than your eyeballs, for example? And really what I'm going to be covering is a brilliant paper. It is my absolute favorite paper, and I'm going to be going into details because it's how we discovered the age of our tissues. Quick point here, right? Let's take a brain cell, right? The brain cell, it's got a membrane, it's full of liquid, it's full of salts, it's full of proteins, and it's full of DNA, right, inside your brain cell. The membrane, the water, the salts, the proteins, the sugars, all of that kind of stuff changes regularly throughout your life, right? But it's still the same cell, right? You consider yourself the same, even though your skin has changed, right? That is still you. So it's still the same cell. It's just that parts of it are constantly being replaced. However, the DNA in there doesn't get replaced unless the cell divides, right? Then new nucleotides will be incorporated into the DNA as the cell divides and divides and divides. That's the only point really that a substantial amount of nucleotides are incorporated into the DNA. So that's the only time that it sort of gets replaced. So when we're talking about how old your tissues are, we're talking about cells, and we're talking about DNA within those cells. So how often do you get new DNA, which is an indicator of a new cell being formed, right? So if it's the same cell that's been sitting there your whole life, the DNA is exactly the same, but some of the other parts of it have been replaced, especially the water, right? That's flowing in and out all the time. Okay, so that is the definition of how old your tissues are, and that's what we're going to be jumping onto in this video. So this is the name of the paper, Retrospective Birth Dating of Cells in Humans. I really think scientists need to learn how to sell things because that's quite a drab title, but the research of it is fantastic. So shout out to Spalding of the Karolinska Institute in Stockholm. This is amazing research. So let me jump into it. Now, it literally starts with a bang. That's how good this paper is. It relies on the fact that we did above atmosphere nuclear testing. So this is a nuclear bomb going off, and this whole paper relies on the fact that we did this, and we essentially made our atmosphere radioactive. So let's have a look at this. This is the graph of the number of nuclear explosions that occur. It's kind of colored by country. Now, red is the Soviet Union and blue is the United States. So you can see they're doing all the testing pretty much. The x-axis is the date. So along here, we've got the date. And if the bar goes up, that means a lot of atmospheric nuclear testing. And if the bar goes down, that's a lot of underground nuclear testing. Now, you can see that there's this time point here, 1963, where all the nuclear testing goes from above ground to below ground. And if we zoom in, that's because there was a test ban treaty that banned atmospheric testing between the major superpowers. There was still the odd above ground, but it put a limit to above ground nuclear testing. Right, and that was in 1963, an important date for this paper. Now, another important principle that we have to cover in this paper is radioactive isotopes. That is, atoms can live in different forms, and an atom is defined by the number of protons. So all carbons have 6 protons. If it doesn't have 6 protons, it's not a carbon. But the number of neutrons, the neutral charged particle in the nucleus of an atom, differs between carbon types, and these are called isomers. So there's carbon 12 and carbon 13 that are quite common, and then there's carbon 14. This is actually a radioactive form. It's a radioactive isotope of carbon, and it has 8 neutrons. It has a few extra neutrons. Apparently, I'm not a physicist. That makes it more unstable, so it breaks down, and that is radioactive decay. But it's important that it naturally decays very slowly, and it decays into these other carbons, basically. And it takes 5,000 years, over 5,000 years, for half of it to decay away and turn into the other forms of carbon, right? So it's a very slow process. That's a very important point, right? So carbon dating is when we look at organic matter and we look at the ratios of carbon 14 to carbon 13 and carbon 12, and we can date things within the thousands of years range. So that's looking for very old, and that's based on radioactive decay. What we're about to talk about is not dependent on radioactive decay. That's a very important point, right? It's only dependent on the fact that we created a lot of carbon-14 when we did those above ground nuclear testing. And that's what this graph is here. So this is atmospheric carbon-14 concentration over time. Here we can see it just chugging along. And then we see it shoot up massively in the 1960s because of late 1950s and early 1960s because of that atmospheric nuclear testing, releasing radiation into the atmosphere that created these radioactive isotopes of carbon. Then the carbon-14 declines. It declines rather rapidly in a predictable, smooth way as on its way to the present day. But didn't I just say that it only decays, half of it only decays every 5,000 years? This looks like half of it decayed in about a decade or so. What's going on? The key here is this is not radioactive decay. The C14 isn't disappearing because of radioactive decay. Well, where is it going? Well, it's going into the biomass, right? Plants take carbon dioxide, which contains carbon and C14 carbon, and they incorporate it into their body and into their trunks of the tree and into the leaves, and then animals consume that, and so then we also absorb that C14. It's also getting soaked into the ocean. That is another huge sink of carbon dioxide that soaks up a bunch of the C14. The key thing there, though, is that atmospheric radioactive isotopes get incorporated into vegetation, which gets incorporated into animals, and we eat plants and animals, so it gets incorporated into us. So essentially, we absorb the atmospheric C14, the radioactive isotope that's in the atmosphere. So I'm just going to give you a quick summary of what the goal of the paper. This is what it intended to do, and then I'm going to tell you how it did it. So we know that the C14 follows a shape like this. It goes up in the atmosphere, then it declines. It's a little bit warped. I drew this, sorry. Yeah, so it goes up like this, and then it declines in a predictive way. Now, because our body got the carbon from the plants and the plants got the carbon from the atmosphere, when we are born, if you were born in 1960, when you were born, you had this amount of C14 in it. This is the theory that the paper is working on. So when you were born, you had that amount of C14 in the DNA in your cells, right? So your DNA contains the same C14 percentages as the atmosphere because ultimately that's where you got it from, right? And then if we look today at someone who was born in 1960, they've got a bunch of organs. What if we checked to see how much C14 was left over in their organs, right? Because how much C14 was left over in their organs from when they were born tells us when the organ was fully replaced. So an example might be, we might think that the brain isn't replaced very often, right? Perhaps the neurons aren't replaced very often. So maybe the C14 levels will be equal to a much earlier date because that's when the cells of the brain were replaced, right? Because ultimately we would replace those cells by eating food, getting nutrients. That C14 would then go into replacing the cells in the brain. Whereas our intestines, we might guess, are constantly shedding their epithelia and constantly being replaced, or perhaps our skin cells, because we can just rub our arm and skin cells flake off. So they're obviously being replaced. So they're being created today. So they should have the same C14 as today. So this is the principle. Ideally, this is the theory that the C14 levels in the DNA in your organs should correlate with when they were last replaced. Because when we replace them with the food that we ate, and the food that we ate ultimately got its carbon from the atmosphere, right? So we should be able to draw this beautiful correlation, aging our organs, figuring out the age of the organs. So that is the theory. This is the idea that they came up with. They were like, let's have a crack. This might work, right? So this is what they did. Now they had to be rigorous, right? They're scientists, right? So they had to be rigorous. Step one, they needed to find out the exact C14 levels since 1950 of every year, right? So they needed to think, how do we figure out what was the exact C14 levels every year, right? And I'd like you to just pause. How would you do it? How would you do it? Okay, here's a hint. This is Tane Mahuta, a very old and large tree in New Zealand, very famous. Now, trees grow in rings, and these rings are due to the seasons, the wet and dry seasons. So each ring represents a year, right? So we can actually count back the years in that ring. Now, those rings were created from the carbon dioxide in the atmosphere. And the carbon dioxide is the radioactive form of carbon. It includes that radioactive form of carbon, C14. So theoretically, if we go back and look at the C14 levels of each ring in that tree, we can calculate the atmospheric C14 levels precisely because they would have been incorporated into each ring in the year. And this is exactly what they did. So they went and they got a slice of tree. Now, in science, we like to confirm things, right? So they wanted to confirm that. They can't just rely on a few trees. So they went and they got Arctic ice cores. Now, because new ice is laid down every year in the Arctic in a predictable way, in a well-mapped way, you can take a big core of ice, and the deep ice represents old and the top ice represents new, and as it freezes it captures little bubbles of gas, and that gas is from the atmosphere. So you can measure that, and that perfectly correlated with the wood. So they've got an excellent confirmation that they have an accurate description of the C14 levels in the atmosphere for decades, right? So now they've got the reference material, they've got that graph that they now wish to read off. The second thing is that they need to validate that at birth, your tissues contain the same 14 levels as the atmosphere did that year. This was a much harder one, and it actually it relied on something quite sad, really. What they did was they used stored frozen blood and tissue samples from infants that died soon after birth. And because those samples have been frozen for over a decade, like 15 years, they could then check, did they have the C14 levels equal to that year? So is the idea that you're born with the same C14 levels in your DNA as the atmosphere, is that true? Because if that's not true, you can't do the correlation, right? So they had to check that. And these kids died of sudden infant death syndrome, which is an absolutely tragic syndrome. And what they found was, yes, the samples that they had the exact same C14 levels as when they were born, which was in the late 1980s, right? So they've confirmed their baseline. When you are born, your DNA contains the same C14 levels as the atmospheric carbon dioxide does. Step 3, validate that new cells, so if I was to create a new cell, has the same C14 levels as the atmosphere right now, right? So the current atmosphere right now. Now, to do this, they use neutrophils, and I covered this in a previous video, actually. Neutrophils have a very short half-life, 24 to 48 hours, right? So they don't live very long. So we know that if you find a neutrophil running around your blood, it's pretty much a brand new cell. So they extracted the DNA from neutrophils from older patients, so very old patients, and they showed that the C14 levels of the DNA of the neutrophils was equal to the low levels of the atmosphere taken now, and not the high levels seen in the atmosphere when the older people were born. So graphically it looks like this. The subject was born here. What did the DNA of his neutrophils contain? It contained the C14 levels of the day that they were sampled, right? So we've totally got both ends of the scale covered now. When you're born, you had the same C14 levels as the year of your birth. And if a cell is created now, it has the same C14 levels of today. So we can assume that if a cell, like a muscle cell, was created 10 years ago, it should have the C14 levels of 10 years ago, right? So they've totally validated the pipeline. It's brilliant. Now they need to do is test the samples, right? So step 4 was sample tissues from a range of places over the body, and measure the C14 levels, and then correlate that C14 levels with atmospheric levels, historical atmospheric levels, to discover the age of the tissue. And this is what they found. So they used people who donated their bodies to science, and they found that the cerebellum, which is a small part of your brain up the back here. It has C14 levels very close to the C14 levels of the birth of the subject in their DNA. The cortex, which is the outer regions of your brain here, was a little bit later, suggesting a few more cells were replaced, and the intestine was down here. Now, this was surprising because we don't think neurons are being replaced very often. But remember, what they're doing when they do that is they extract all the DNA from that part of the brain. But the brain isn't just made up of neurons. It's made up of a bunch of other cells, right? Including an innate immune cell called a microglia, and also support cells called astrocytes and glia, right? So what they wanted to do is then split out the cells, take the neurons, have a look at the age of that DNA, and then take the other cells and take a look at the age of that DNA. So cortical neurons were almost the exact same age as birth, right? So the DNA in your neurons is the same age as you are. That molecule, my molecules in my brain cells, my neurons, are 35 years old and they haven't been replaced. It's amazing, right? What I love about that, and I don't want to get a bit wishy-washy, but if you were to ask what biologically defines me, I would say my neurons, because they make up my personality, my memories, even my decisions, everything is encoded in my brain. So my neurons, and I would say my DNA. My DNA has the blueprints for building a jack. So those two things, and then the DNA in my neurons hasn't changed since the day I was born. It feels like, there's a continuous thread. It's almost spiritual, but it's not spiritual because it's molecules. I love it. It's amazing, right? So yeah, the DNA in just your neurons pretty much hasn't changed since the day you were born. There's a teeny tiny bit of neurogenesis in some parts of the brain, but pretty much the neurons that you're born with is all you've got. It's amazing. And then they showed that the non-neuronal part was much younger, but still not amazingly young. So the astrocytes and microglia, this person was sampled in 2005, and the average age of the astrocytes and microglia, the non-neuronal cells and oligodendrocytes and stuff like that, is sort of mid-1970s, right? So the replacement rate isn't huge in that region. They found that muscles were about 15 years old. Isn't that amazing? Your muscle tissue is about 15 years old. They found the intestines were 10 years old. Now that was a little surprising because wisdom is that you're constantly shedding your epithelia in your intestines just like you're constantly shedding your skin cells, right? That's the general understanding. But you got to remember they mushed up the whole intestine. And so later research basically showed that the epithelial cells are replaced almost daily, whereas the basement membrane and smooth muscle and all the other stuff that goes into it, the blood vessels, it's a bit older. It's probably 15 years old. So once you average that together, and there's a lot more muscle and basement membrane than epithelial cells, once you average that together, you end up with about 10 years old. And they found a similar story with the skin. So the epithelial layers are very young, but the basement layers are very old, and so once you average it together, you end up with about 10 years old. But it seems like your skin is weeks old. So your skin is weeks old. Your skin is weeks old. So this should be a little bit of a wow moment, right? If your skin, your outer skin is weeks old, and even like the basement support layers and stem cells and all that stuff under your skin is 15 years old, maybe 10 years old, right? That still means that your skin and my skin is the same age as this guy's skin, Donald Trump, right? And I don't moisturize, but boy, look, not a wrinkle when I'm 35. Come on. I mean, okay, there was a wrinkle. Ignore that wrinkle. There's maybe only 1 or 2 wrinkles, but my skin looks all right, okay? I don't want to age. Anyway, my skin looks okay. It looks much better than Donald Trump, next to Donald Trump. Look at that. Oh, so smooth. So my skin looks way better than Donald Trump's skin, yet they're the same age. So what is going on? And in fact, why do we age then? If my skin is brand new, why does it start to look old? Why does Donald Trump's skin, even though it's only maybe 10 years old all the way through, 15 years old, why does it look so old and wrinkly and disgusting and horrible? Donald Trump. Anyway, And I think this should break a wisdom and intuition that you should have had about aging. Most people think humans age just like cars age, right? They get exposed to the environment, the parts wear out, they break down, and they degrade, right? That's aging, right? We think the intuition is we age just like a car ages, just like a house ages, just like a mountain ages, right? It erodes, right? A mountain is constantly eroding. That's the intuition on why we age, but it's completely wrong, right? Because a better analogy would be, what about a car that you replace its parts, all of its parts pretty much every 10 to 15 years, right? So like the outer layers of the car are replaced, regularly, right? So if we did that to a car, it would never age, right? If we constantly restored the car, it would never age, and our bodies are constantly being restored. So why do we age, right? Your intuition about aging is wrong, and that's where it's really going to get interesting. So in the next video, I'm going to discuss, why do we age? Why? The molecular mechanisms behind it, and maybe some evolution as well. Really good. So check it out.